Okay, welcome everybody. It's my pleasure to welcome Magdalena Ivanova.
Yeah.
She's a research associate professor in neurology,
Yes.
and she has an exciting project that could potentially use some molecular modeling. And thought it'd be a good opportunity to see what like a target application for drug discovery and other discovery projects might be. So
yeah, thank you so much for the invitation, and thank you all for coming. And this it's kind of my pet project. It's just we've done a little bit of it, but your help will be really welcome. My background is in structural biology. This is how I started, and I was working as my postdoc on solving the structures of amyloid proteins, and a large number of neurodegenerative diseases are caused by accumulation of amyloids of these diseases. And this is how I end up being in the Department of Neurology, primarily studying these neurodegenerative proteins. I got really excited about this particular one because it's in contrast with the neurodegenerative ones, which are mostly natively unstructured. So it's just trying to do like any drug screen and anything like that, it will be really very challenging. This one has a structure, and it's expressed in the cornea, and this is where it aggregates in the corneas. So I really love before. So how I just advanced is that I need to click on that. It's the same. All right, and this is checking it works. So I really like to spend a little bit of time, and I really there's the clock. A little bit of time just giving a background. This protein is involved in the so-called TGFBIp related corneal dystrophies, and as you're guessing, mutations in this protein, and all of them are listed here on the left, associated with a wide range of dystrophies. And this is the abbreviations which I'm using because it's a mouthful spelling out the whole protein. It stands for transforming growth factor beta-induced protein IGH3. So I'll be using TGFBI from now on. So these dystrophies are autosomal dominant, meaning that if you get the mutated gene, you will get sick of it. And it is one other interesting thing about that. It's maybe if you have not heard about that, but this is the epidemiology of the corneal dystrophies. There is a quite large occurrence in Europe and Asia, some in like North America, and about 9 to 15 people in every 10,000 will have that condition. This is leading all these people who have that mutation, they have a loss of vision with the time, and this can start in juvenile age or later in the time. Also the wild type protein, just keep on going for the importance, is involved in the so-called Fuchs dystrophy. And in this Fuchs dystrophy, there's again a loss of transparency of the cornea. This is the very front layer of our eyes. And in about 4% of people which are aged older than 40, they will have that type of dystrophy, and it's also keratoconus. So it is not very specific for this corneal dystrophy diseases. This is where is the mutated form, but also the wild type form has quite a bit implications in disease. And I want to, I guess, yeah. And here is just to show, this is the amazing thing about this type of mutations, because here are shown all the different phenotypic manifestation. They all look different. The corneas, they are all with a reduced transparency, as you can see, but the accumulations, these whites out things of the proteins are they all have different types. And some of them are like present like a halo, the other ones are like fibrillars and so forth. And I just want to pay attention to this particular mutation, which is involved in the classic lattice dystrophy. When you have it, a mutation of arginine 124 to cysteine, it is linked to the accumulation of amyloid aggregates. So this is where is my expertise is coming. To give you the this is a dissection of the cornea. It is kind of a complicated structure and has different layers, and starting from epithelium, going to endothelium. And here I'm showing the accumulation of TGFBIp aggregates are occurring. It's on the top layers of the cornea primarily, but Fuchs dystrophies is all on the bottom. So what are the current therapies for these diseases? They are mostly surgical. The first one is phototherapeutic keratectomy, or PTK. This is like the physicians use a laser to shave off this cornea. And like, for example, in most lattice dystrophies, for example, TGFBI is accumulated on the very top layer. If you shave it off, so you're going to reduce the accumulated protein, so increasing the vision and the transparency. The other one is the cornea transplants. This is in a case when the accumulations are deep in the cornea, so you cannot access with this laser treatment. There's numerous problems with these treatments. The biggest problem is that condition reoccurs in about 5 to 6 years. So all of this needs to be repeated. And cornea transplants, they have a certain limitation how many times you can get them. And currently there are no medicinal treatments which we know of, which are available for this type of condition. So our hypothesis and what is it's centered, it's very directly linked the causative effect of TGFBI, that it's when it's aggregates, it causes the ossification of the cornea, and this in turn is causing the loss of vision. So one, you can imagine if you stabilize the native structure, this is in the case of the mutants, or if you prevent or try to target the aggregation of this protein, you would expect that you can alleviate this condition. So we will be going to talk a lot of structure. Your structural people modeling the structure of that protein, which is 683 residues in length, has been recently solved. It has this banana shape, just for you to let you know that the very C and termini, they are very flexible. You cannot see it on the structure. Notice that here in the middle, there is this FAS domain, and 2 and 3, they are connected through a flexible linker. So there is a lot of movement occurring in that area. So this is something which one might consider for stabilizing. As the function, it's mostly involved in the cell adhesion, and this is extracellular protein which interacts with integrins and collagen in an extracellular matrix. Just a side note, it's also been implicated as a suppressant promoter in cancer. So, so this protein is extracellular. So again, it's very easily accessible if you want to try and test drugs on that target. So I just briefly want to give an example of how you can handle the case of the stabilizing of the proteins, and just to give an example that's possible to design a chemical chaperone, and this in the case of the transdirectin, which is amyloid forming protein. And in fact, it's kind of interesting that all people of old age, all centennials, actually, they die of amyloidosis caused by this protein transthyretin. On the side note, that in the native form, the transthyretin exists as a tetramer. However, if it's for whatever reason, this tetramer is being destabilized and it dissociates to a monomer, then a monomer quite readily can start aggregate and form spherical and amorphous and fibrillous aggregates. So if you use a molecule to stabilize it, in this case, you see that black molecule, which is called tefamidis. If you lock that tetramer, then you have a reduction of this aggregation of this protein. And in fact, this has been used as a drug to cure the transthyretin, the amyloidosis caused by transthyretin. So example where there's a possibility for like a target where you can try to design a chemical chaperone for this particular protein. Of note, TGFBI forms multimers, and the formation of multimers, dimers, tetramers is really dependent on the concentration of the protein, but in the cornea, it's very high. So it's likely exists in multimeric state.
So how much about the multimeric structure of the GFBI? And does it fold switch when it goes into the amyloid state?
So there is no structure of the amyloid fibril. That will be wonderful. I've been really wanting for a while to kind of try to do amyloid cryo, because right now the only way to solve structures of amyloid fibrils is doing a cryo-electron microscopy, but we don't have the expertise. I mean, not that we don't have the expertise, but you need to have someone who works with fibrillous structures, or like someone willing to learn to pipeline it. But yes, that will be really beneficial to know what is the structure of it. I'm going to talk in a few minutes. There's a way to predict the regions which might be involved in the amyloid aggregation, and this can be screened to kind of yes and no whether they are involved in the amyloid formation.
So
Question. Is the transthyretin part of the amyloid deposition, or is it a totally separate protein from the beta amyloid? And
it is a completely different protein. So that's the confusing part. Many proteins can form amyloids. So transthyretin is one of these proteins can form amyloids, and the hallmark of amyloids is a beta sheet formation. So there's a conversion from the native structure of any of the protein. It can be transthyretin, can be amyloid beta, or any of these proteins. They has to have a conversion into the beta sheet, and I'm going to show a little bit of that coming.
thank you.
So you also indicated they form native multimers, right? So non-amyloid. What do we know about those structures?
This is in like when they're concentrated. This is they form, but I don't know. We just have the X-rays like structure, and I'm not sure if it's a monomeric form or the dimer on the structure. I need to go back and check.
Thank you.
But it's really dependent on the concentration, so that will be really hard to pinpoint. All right. So how am with the time? 10. Oh well, but we are discussing, right? So I can go. All right. So I really want to point here one thing which I like to show when I speak about the protein structures and aggregation. They're kind of two sides. For when you have a native formation of the native structure, it's the folding of the nascent, freshly synthesized nascent chains of the proteins goes through many energy minimum, and in the end, it just gets into this its native fold. And these are mostly driven by intramolecular contacts and interactions. This is within the polypeptide chain. However, in all these other sites on the right, there is, when you have any multimerization, which is the oligomerization, the amyloid fibril formation, and now it's been really coming up very strong, the phase separation of the proteins. All of these processes are driven by intramolecular contacts. And typically large proteins, including TGFBI, they need to have the help of the chaperones. So you can imagine if there's any mutants which kind of misfold and cause a regional misfold of the protein, or like downregulation of the chaperones in the cells, there would be a lot of these quasi-folded forms of the protein would linger longer to have time to actually start interacting with other molecules, causing that abnormal aggregation, which is actually not good for the cell. And this is my slide pointing out that amyloid formation and deposits are found all over the body. It's kind of a nice map. We really are aware and know quite a bit of the aggregation of amyloid beta, tau, alpha-synuclein, at least I'm working in that department. However, I want to point out that TGFBI is the protein also forming amyloids, and alpha-beta crystalline, the protein which aggregates, and it's causing the cataract formation in the old age. So all of these, and I already mentioned trans-tyrosine, but all of these proteins end up, as I mentioned, going through this structural change in which a lot of different subunits come together to form these beta sheet rich structures. And they are also, if you do an X-ray diffraction of them, there's a very characteristic X-ray pattern, and for the long time it was known that it's the amyloid fibrils, so these particular aggregates very rich in beta sheets. But there is a large conformational conversion in the case of this protein. So what is going on? I kind of was mentioning most of the proteins, when they come to interact with one another to form these beta structures or amyloid fibrils, they interacted only through a specific regions. It's only a pieces or segments of the proteins, they have this propensity to interact with one another. Because everybody who works with structure and looks into the structure knows that stretches beta sheets because of the twisted pattern cannot be longer than maybe 13, 15, maybe I'm old, a stretch of 13, 15 amino acids. So all of these segments, which when they get exposed due to the misfolding, they come, they can interact with one another and like form these fibrillar structures and beta sheets. All right, so coming back to TGFBI, they are actually aggregation predictors. And below a map in red, the segments within TGFBI and in red, and also you can see it's situated all around the structure, the regions which all predictors and consensus that they can drive the aggregation or amyloid fiber formation of this protein. And here are just given the regions in the red spots, and notice that here, like in blue, I have pointed out the places of where most of the mutations for these proteins being clustered, there's more than 70 mutations of it, but many of them are either clustered in this region around 555 or here around 124. And notice in both cases, there's an enrichment of these amyloid segments. So if you have this original misfolding, you are kind of increasing the propensity of this, the likelihood of these segments to come together and interact into aggregates. So what is the strategy in this case? If you know which are the regions which are responsible for formation of the spine, beta sheet spine of the amyloid fibrils, if you design an inhibitor which binds tightly to this region, you will prevent the other molecules from coming to interact. You're literally capping or preventing the growth of the fibrils. And in fact, there has been a really successful, at least in vitro studies showing that if you design inhibitors which bind on the top of the beta sheets on the sides, or like which have bulky residues to prevent this, like the assembly of the mating of with the other beta sheet, you can actually disrupt the amyloid fibril formation. The problem is typically like this can be designed using the structures of the peptides. I've really solved a lot of it. You can actually take all the predicted peptides, validate them, and they really show that they're a good target for, they're indeed involved in the amyloid fibril formation by solving the structure of these peptides, or like this is a different route to having the structure of the full length. Then you can use that structure to design a peptide which will prevent that extension of the amyloid fibrils. So here I'm going to skip it because of the restriction of the time. What I want to point you, at least in the case of other amyloid proteins, we don't know how it is. We just need to be aware is the complexity that there's many different ways the molecules can self-assemble and into the amyloid fibril. So this is something which we need to be aware. And now coming back to the little work we have done with this protein, we were able, so this one is not good for expression in E. coli. It has a lot of disulfides, so it's only can be expressed and purified from freestyle hack to 93 cells. And we were able to purified, and here is the gel. The protein has a HISTAC for a nickel NTA column purification. And of a note, like there is an assay which can actually detect, and we can be used for screening whether the amyloid fibril formation is occurring or not. It's a molecule, which is thioflavin T, which produces a fluorescence only in the presence of beta sheets. So if there is no beta sheet, so there's no amyloid structures, there is no signal, no fluorescence signal. But if the aggregates, amyloid aggregate starts to form, there is increase in the fluorescence. So it can be used in case like any targets it's found against that protein, and especially in the case of this R124C, which is the amyloid forming protein, can be used as an assay to screen whether the compounds are actually active and can prevent the aggregation or not. And I really want to acknowledge this work has been done with 2 undergraduate students, and we had a very small seed grant. Just before I leave here, I was very optimistic about purifying that protein since the crystal structure is being solved and published already. Off of note, it took quite a bit of time optimization to get it started, and growing, like expressing proteins in eukaryotic cell, it's a very, very expensive enterprise, both in time and in regions like us. So we are still needs to optimize it a bit more. And this is the end of my presentation. On that top corner, you can see I've listed the possible culprits of protein aggregation, and I kind of touched base on some of them. Here is the possible ways, and in green, I took the liberty to highlight the ones which can be or may be accessible for computational design of inhibitors or screens. So this is inhibition aggregation, stabilizing of the native fault. And I also want to list you that other tools which can be potentially available for screening the different molecules. Animal model does exist, which is really nice. And I also want to acknowledge my collaboration with Dr. Nalasamy, who is a corneal surgeon, and he can provide ex vivo corneal tissues for screening compounds, which, for example, if any of them are isolated through this theoflavin T aggregation assay. So thank you all. It's 3:30. I don't want to keep you longer, but I'll take a few questions. Can I? Or
Let's take time, maybe we'll take questions
after, yeah, maybe.
While we're doing set.
Oh, okay. Yeah, yeah.
The last slide is that explaining
Is it on here?
They're both negative, but sometimes it really depends on how much on the way the samples are processed. You add during the last session, you really have to
Although I am.
It is actually on the slide, but it depends. If you ask me in vitro, it's I believe it's a couple of days, but just if you use the purified stuff. But if you but in cells, I don't think we have the assay to actually trigger the aggregation of these proteins itself, but it is in this extent. But it's the secreted one, so it will be a little bit more tricky to come up with tests.
Is for modulators, is this meant to target the wild type or would properly call it, or just the misfolded?
I think we should target both, because there is a reason to target these like both, and in fact this is the way we should kind of proceed. These are things, they don't work, at least in the case of the accident properties. At least that's my belief. But for the case of isolating candidates, you should do it one.
Okay, let's take Magdalena one more time. Okay, for our next talk, I'm thrilled to have Junzhe Guo.
Oh, yeah.
He is a visiting researcher and working in the Freddolino lab with Chengxin. And he's going to tell you about ultra-fast loop modeling for atomic RNA structures.
Thank you for introduction, and thank you for everyone to come. My project's about RNA loop modeling. As everybody know, wait, how to move? Oh, everybody know. Okay. Everyone knows RNA plays very essential role in many biochemical events like splicing, transcription, translation, and our structural analysis of RNA is very important for us to understand their biological function. And like this is a structure of human spliceosome, and it's very essential for us for understanding about splicing mechanism. And for most RNA structure obtained by crystallography experiments or cryo-EM, we always find some nucleotide missing. For example, this is a crystal structure of a ternary complex, and this dot line means missing nucleotide. Though they might be not important for this structure, but for some, when it matters, will be a big mess. It will be a big problem. And now we don't have very efficient loop modeling program, and now the most efficient one is PDB fixer, but it's still very slow and inaccurate. So my program is about developing a C++ program to reconstruct RNA structure with missing nucleotide from an atomic level. And
I think I'm sharing the wrong screen. Let me fix this. We'll get this set up. Thank you.
And globally we have RNA. RNA is the deterministic program to reconstruct nuclei of any identity with at least one atom. As long as the RNA structure have at least one atom per nucleotide, it can reconstruct the full structure, but it can help with structure without any atom in it. So it can be used as an optimization algorithm instead of a program to generate initial configurations. And here's my experimental design. First, we need to obtain some RNA structure into a database to benchmark, and then we employ different methods to generate RNA initial configuration, and then we incorporate some package to optimize the generated structures. And this data set obtained from PDB, and it consists of over 600 non-redundant RNA structures, and most of them are actually a single chain, and they don't have any non-standard nucleotide and easy to perform by CDP. And here I will introduce some generating method, and here is a very vague illustration. I will detail it later. And oh, okay. Sorry, I'm a little nervous. Please forgive me about that. And first method is about linear field. Suppose this structure is the real structure, and we need to generate something like this. And this is input what we know from experiments. And here you can see a long gap in them, and we need to put 4 residue into them. This circle means residue, and this sticks mean the length between suppose there are bonds between them. And for linear field, we just draw a straight line and put these residues into them. And if you compare to the real structure, you can find some of them may be very accurate, but most of them is not accurate, and it's very simple and also very inaccurate. And if the gap happens on the edge of the RNA structure, it cannot take the problem. So there's another method called extrapolation. Still, we put an input like this, and we extrapolate from these residues and this residue, every 2 for each end. And you can find for nucleotide just adjacent to the existing structure, they are very accurate. But for a longer gap with a longer distance between 2 ends, these residues will be very inaccurate. And also it can help with edge cases. And here is the third method about spherical random sampling. Spherical random sampling means I will generate a residue based on the age of existing structures. It will set the massive center of this residue and the spherical center, and I will put new residue onto the spherical surface. And for the first round, they will put these three residue, and the next round, we can fully reconstruct the structures. Though it's very fast, but sometimes very inefficient, because if you have this gap really long and it doesn't have too much residue to put in them, it will just look like straight line and will take much longer time. But these cases will be easier for linear field to complete. So I think when employing this method, I need to incorporate linear field. And also we can have some combined methods. These combined methods employ both linear field and extrapolation, and sometimes have better results. For example, this combined method, the first step will generate a residue with extrapolation, and we'll compare the distance between new generated residue and the other end of the structure and one end of structure and structure. If the distance is shorter, then we adopt the new generated residue and we kept generating others. And this one is also closer. The next one is also closer, but this one is a little far away from where it should be. So we just abandon this residue and generate another one with linear field. And it looked much better than both extrapolation and linear field. And for other combined methods, there are two options. Both we need to extrapolate from the existing residues, and we'll compare the distance between new residues and the existing structure, like 0,4, and also we will compare the distance between 1 and 3. And for combined method 2, if we find the distance fitable is fit for residues, and we will adopt them randomly. But sometimes we can find this cases, so there is combined method 3. And here we combine both 1 and 2, 1 and 1. And for residue between them, we use a linear field to fill them. Sometimes have better results, but in most cases, it will be worse than linear field.
Just for clarifying for people in the room, which atoms are you actually reconstructing in these methods?
Okay.
So you have missing residues, right? But you're showing a set of points here.
Yeah, have missing this. So we'll first generate eight atoms, and then it will compare the distance between 0,4, 1, 3, 2, 2, 3, 1, and
I'm sorry, more generally, for all of these methods, are you doing just the phosphorus atoms? Are you doing some other one atom per residue?
Actually all atoms on the back
All atoms on
bundle.
the back,
Back bundle.
and then you can optimize.
Yes. So let's move on to the next slide. Still there's many secondary structures we can usually find many secondary structures in RNA structures, and here is an example of RNA. It has many secondary structures. If one of them is lost, then we can easily reconstruct them with base pair. So I adopt some dot bracket notation file to generate this helix. And in dot bracket notation file, this bracket mean a base pair. For example, this string will base pair with this string, and they can form a loop. And so I'll go on to the loop non-loop base pair. If we have already know there is base pair between the existing structure and the loop we want to generate, then we can put them to make the text more easier. For example, this green loop is we about to generate. and this colorful structure is what we already have. Then we can first put some residue like this to make the text more easier. And after I generate all the initial configuration, I will optimize with RNA. RNA mainly improve the bond length, angle, atom clashes, and base conformations. And here are some examples. If they find the balance too long, they just shorten them. If they find angle is too big or too small, they will adjust the bond length between them to make the angle looks better. And because I usually imply linear field to generate configuration, so that's common that many atoms are on a straight line, and if you just shorten the distance between them, this atom just be pushed on this straight line and never get changed. So I still need to adjust the distance between the adjacent residue between it to make the angle looks better. About these many methods, and how about the RMSD results? Here is some RMSD result with all these methods. From this, you can find the linear field, spherical random sampling, and combined method. Sorry, I make a mistake. Actually, this one is the third best method between them. But how we could imply them to tackle the real problems? Because if the gap length is relatively large and the residue is relatively small, linear field will perform better and spherical random sampling will perform worse. But for the contrary cases, spherical random actually perform better. So we need to find out the relationship between the generating methods and the gap length or the gap size. So I make a big figure about it. Well, it's hard to read. So first, I want you to pay attention to this red box and yellow box. These two blocks mean extrapolation and combined method too, and they generally use extrapolation to generate figures. So when the gap size is really large, extrapolation can perform very well because just tangle them. The residues adjacent to the existing structure looks good, but for the residue in the middle, they will not very good. And for small gap, combined method will usually perform better in these cases. The green blocks, they usually.
Are they like?
Oh, wait. Sorry, I beg pardon.
How significant are the differences when the error bars are overlapping them?
Well, I think this part about RSD, actually, I don't know much about how there's difference between them. But in these cases, I think the linear field and combined method one they don't have a very significant difference. And okay. So for larger gaps, linear field actually perform very well. That's very surprising because I usually think the linear field is very simple and inaccurate in most cases. Actually, I don't. It's a very surprising finding. So for future work, I will work on to imply loop base pairing, because if there is base pair in the generated loop, then if I can put them in the right position, the text will be easier. And I will always I will also use OMM package for further optimization. For example, this is a structure optimized by RNA. It has a very big mass here. It should be a loop. Yeah. After performing OMM, actually we can find this a little messy, but still very careful. Compared to the real structure here, I still need a lot of work to do. Yeah. So that's the end of my presentation, and thank you everyone for hearing.
Yeah. I am wondering, so a lot of times when loops are dissolved in structures, it's because they're more linear.
Mhm.
So I'm wondering if you account for like different structures.
Yeah, this is, yeah, I have the same. It's like, can you actually have, multiple confirmation with the same score? Because if they're like, structures,
Yes, they can be possible. Yeah.
because, but you're fixing it one. How you select that one? I'm sorry.
No, I just randomly selected. I didn't compare much about them.
But does their algorithm gives you fully good scoring other options? This is-
Yeah, just I didn't work much on the selection part. So now it's just a comparison.
So initially you're doing like a geometric
Uh,
identification, and then
yeah.
you're adding a force field to like minimize.
Yes, I'm working on adding a force field on that, but now it's still very image. Thank you for your question. Was that the question?
Yeah.
Oh, so I can I just quickly add then, I mean, I just, because there's a slight mismatch, it's not just a minimization that's being done with the force field now, but actually implicit solvent, molecular dynamics, which also gets this question of multiple confirmations, because depending on how long we run that and whether we do some tempering or anything like that, it might be possible to generate multiple confirmations that way. It's something that needs to be explored.
So geometrically, what are you trying to optimize then? How do you know that, you say that they're not going to be on a straight line. So what is the function that you're trying to optimize or to find a solution?
You mean the this size? About that. Yeah. Just if you find the distance between them are too long, then we just shorten the distance, move to here. And we also compare distance between these two residues. If they come, they will move it to a shorter distance place.
So this is based on deviations from known bond lengths, right? And geometries?
Yes, the bond line obtained from an ideal RNA helix.
Maybe I missed this context in the beginning, but is the idea here is that if you just randomly put atoms in the loop, it's not a great starting place for, say, MD or minimization. And so you're trying to come up with something more plausible that you can
Yeah.
thread initially that will lead to an ultimate better solution. Okay, so it's like not directly optimizing the RMSD of the atoms because straight line isn't real, but it's something that's somewhat plausible. So that way you can then feed it into a simulation and it won't like totally blow up eventually. Is that sort of?
Yeah, I think that's fair. And if you go to your box plot, oh, sorry.
Don't you bias it? Okay, if you like the location, you will be biased.
Yes, there will be bias. No
Well,
bill.
it depends on the unit bias, I guess. I think that if you can run MD long enough, if you could afford to run MD long enough, and you actually trust that the force field can get the right confirmation, then that's going to be a better answer. But I think often we don't want to run explicit solvent MD for long enough to get that structure, or we don't trust that we'll get the right structure that way. And so in some sense, if you could come up with a starting point using some heuristics or eventually maybe a neural network that is close to the right structure, gives you basically the right backbone, and you're just doing local refinement, you don't have to rely on the force field to be able to get the right answer. And so this is the case, like this is better established in protein folding, I think, where like doing, but a lot of proteins still couldn't be folded using explicit solvent MD calculations, but you can certainly get good structures now using brain-based pipelines. So I think that you can similarly here use the molecular mechanics for local refinement if you can come up with a good way to get a close enough starting point, rather than try to start completely from scratch and sample like we.
And the close enough is based on how well it matches with the data set. Is that how you define close enough?
Well, so in this case, he didn't really, you didn't go into how you're generating your training set here.
Yes.
Do you want to say a few words about that?
The training data set based on this over 600 RNA structures, and we track the distribution of nature gaps length and this nature gap length, and then we create gap in this data set to make a benchmark data set.
So it's essentially you're trying to get, so you take a bunch of structures where we actually know the right answer, you pretend you don't, and then you try to match that as well as possible. And there's bias in the selection of where the deletions are being put in to match where are likely difficult to resolve regions, things with high temperature factors. So what we're trying to target here in the early stages is a known structure, because we started from something we actually did know.
Are there like a large variety of methods between the different structures, or are they quite similar in lengths?
Big divide by
The size of the structures, are there large differences in your training set of how large the structures are?
I think how to define large, actually the largest structure is about 4,000 base pair, and the smallest one is about only 30 base pair. And it's yes, we can find many large gap like 2,000 base pairs in it, but we just can make this large gap in a small structure.
And so then when you're doing the optimization of MD, is this the entire system? Or is there some sort of constraints on
now is the whole structure, and I'm working on to make it more constant.
Yeah, because I wonder if it would make sense to constrain the other components that are to some like this initial simulations just within that region, especially if you have really large systems.
I think I'm just in the spherical random. Can you generate different
yes.
configurations, or does it just give you one sample?
It actually have two options. It can both generate many different loops, or just only one loops. It depends on the need, on the demand of user.
I guess what I'm asking is, if you start with an initial position and it turns out to be a bad one, is in some way to sort of re-roll the dice to find something that won't blow up. This might be one of the few strategies you described, ones that actually allow you to sort of generate a new choice at random.
Usually, if I have a bad start, it will take much time to restart.
I see. So you don't get that many shots. Draw your attention to maybe a paper that you may not have seen, but maybe 10 years ago, there was a nice one in Rosetta called Loop Hash, where you take, it's for protein loop modeling, but essentially you take the start and end positions and you fix one of the ends at 0, and then it creates like a, for every distance, every orientation and distance, you have like a sort of a lookup table of other loops that span that same gap. And those mean that you can, if you find a loop length and orientation, you can sort of substitute in through a fragment insertion from a native like structure to span that gap. And it actually is a pretty good starting place because it all has the right geometry. It has like, you know, the correct bottom lengths and angles and all that stuff. It's not perfect because it could clash with things and do other stuff, but at least it doesn't have like broken geometry associated with it. So I don't know if you have enough RNA and there was consistent enough orientation geometries to populate a hash table of this sort, but maybe through simulations you could generate these things and then sort of look them up and insert sort of fragments of real structures into the loop that span. So it might be another strategy to get semi-realistic loop configurations to start with.
As question was, the select fragment from long TGE is starting. So I think there is a program that trying to do that is still within that this performance is extremely bad, partly because theoretically it works.
Yeah, unfortunately, that's probably not true.
RNA is hard. Yeah. Like that's my conclusion to like everything we've done so far with RNA is hard.
There's a lot of power in RNA, and it's been not being harvested, even as in the filters,
What questions? Yeah, let's think one more time. Two quick sort of administrative announcements. The first is since next month will be like on break, so we're gonna push it back a week. I think to January 8th, if I have my calendar right, but not the first Friday, but it'll be the second Friday. I think.
It says the 12th.
12th, that sounds.
No, January 12th. Yeah.
So then the first 2 weeks. Okay. And the second is unrelated to this monthly meeting. We also have we've been doing an every other week Zoom meeting for diffusion models for protein modeling. And we've been getting the authors of very recent papers to come and present them. And it's really a lot of fun. So I'll send out, I'll put onto the website and that way you guys can find the link to that as well. And it's every other Wednesday at 11. So it'll be next Wednesday. And we're having a group from Mohammed Al-Qureshi's group present gene next week. So we have the recordings from the past couple of weeks, and we've got RF diffusion and some of the other frame diff and some other ones, and also nice background on diffusion models for protein structure. So it's a new hot area, and so if you want to come and be up to date with those models, can go.
I can make one other announcement. We have faculty talks. We tend to have one faculty and one trainee talk in each of these sessions. We have faculty talks for the next 4 sessions, but not trainee talks yet. So please reach out to me if you have any interest in presenting in one of the next couple of sessions. We'd love to other groups start presenting. I think my students would also love to have others represented so they don't get voluntold for every single one. Save them. If you don't do it for me, do it for them.
Yeah, exactly. All right, well, thank you so much for coming, and happy break with you. Okay, thank you. Good to see everybody.